Hi everyone, my name is Dr. Tess. I'm a microbiologist and food safety professional. I work in the cannabis space and I also have a research partnership with CU Boulder in their aerobiology and disinfection lab where I work with Aspergillus. And today we're going to talk about Aspergillus testing in cannabis. I've seen a lot of videos and posts about this topic and I thought that it was important with my experience as a microbiologist, which many of these people are not, and in product safety and doing research with Aspergillus fumigatus, it's important that I add my voice to this conversation and hopefully dispel some of this misinformation that I've seen floating around. So just right off the bat, point number one, there are hundreds of species in the Aspergillus genus. So Aspergillus is the genus name, and then there's another name after that to describe specific species. So Aspergillus fumigatus, for example, is a microorganism. That is a different microorganism than Aspergillus niger or Aspergillus oryzae. So those species are all very different. There's over 200 species of Aspergillus within the Aspergillus genus. Some of those species are pathogenic, which means that they cause disease, typically through the route of inhalation. So you breathe in spores or parts of the mycelium, and that can cause, aspergillosis in your lungs. That can also travel to other parts of your body, like your brain. Some Aspergillus species are toxigenic, meaning that they produce toxins. Those toxins can have all sorts of negative health consequences. They can cause cancer, they can be linked to liver diseases, and we only really test for a couple of those toxins in cannabis testing. There are lots of other toxins that are produced by Aspergillus species. And then some of these Aspergillus species are immunogenic, meaning that they cause allergies, which can also be severe. So allergies are actually the 6th leading cause of chronic disease in the United States. And again, that's usually airborne when it comes to Aspergillus. You're breathing these in, you're breathing in the spores and other bits and pieces of these microbes, and that can lead to chronic rhinitis, which is inflammation of the nose and eyes, sneezing, coughing, having headaches. It can even cause asthma or asthma attacks. It can definitely spur asthma attacks. And asthma attacks are a serious matter. About 11 people die from an asthma attack every single day in the United States. And allergens are the leading trigger for asthma attacks. So we should take allergies seriously when it comes to those immunogenic properties of Aspergillus. And then some Aspergillus species are used in food production. So I've used Aspergillus oryzae to make miso at home. So some of these are used in food production safely. You can still develop allergies against them, so that's important, but they won't necessarily infect you, so they're not pathogens. Now, I know in cannabis we like to group all these Aspergillus species into one big name, Aspergillus, but there's really, really big differences when you look species to species and even within strains of these different species when it comes to public health. So right off the bat, I just want to say Aspergillus, very broad category, and you really need to understand pathogenic, toxigenic, and allergenic, and the risks that they pose to people through the route of inhalation or breathing them in. So most Aspergillus species are ubiquitous. That means that they're commonly found in the environment. However, from an indoor air quality perspective, or IAQ, you'll hear me say, there are professional organizations that have put standards on the amount of Aspergillus and other pathogenic or toxigenic species that can be floating around in your air. The American Industrial Hygiene Association, the Healthy Buildings International, and Environment Canada all agree in their IAQ standards that pathogenic and toxigenic microbes should not be found in indoor air. So yes, these are ubiquitous, but there are standards. In the fact that maybe you will find them outdoors, maybe you breathe them in throughout the day, but you really should not have these bad Aspergillus species in the air you're breathing indoors. This is important because very few growers know if they have pathogenic or toxigenic Aspergillus species floating around their indoor grows. Now, arguably, out of all the Aspergillus species that we talk about and test for in the cannabis space or just globally, Aspergillus fumigatus is the worst of all the Aspergillus species. I work with Aspergillus fumigatus in the lab. It is a designated World Health Organization fungal priority pathogen. It's a biosafety level 2 organism. So I must have special training and certification to research it. And I always have to work with it in a biosafety cabinet. That means I'm always working in a contained space where HEPA filtered air is going into that space and HEPA filtered air is going out of that space. So I don't spread those spores throughout my entire facility. When I'm done working with these cultures, I have to autoclave and sterilize the materials I was working with because I don't want to spread those pathogens around. I don't mess with this organism. It's serious business. And I have colleagues who've had family members die from this pathogen. Now, another argument that I've heard is, well, we don't even know the infectious dose, how many Aspergillus spores or, viable cells are required to cause disease. And yeah, that's true. It's true of every opportunistic pathogen. No opportunistic pathogen has an established infectious dose. So, for example, Aspergillus fumigatus is an opportunistic pathogen, meaning it typically causes disease in folks who are immune compromised, but it can also infect folks who are healthy. So let's compare Aspergillus fumigatus to another pathogen that we test for in food and cannabis, Salmonella. So the infectious dose of Salmonella, which is a true pathogen, can range from 1,000 to 100,000 CFU, depending on the strain and the conditions. So how much Salmonella do you need to fail on final product? How much do you need to test for to fail? 1. Even though the infectious dose is 1,000 to 100,000, you only need 1 CFU or detectable with its DNA. Which is much, much less than the infectious dose. Why is that? Because it's a liability to have pathogens on your product or even in your production environment, something many cannabis manufacturers just don't quite grasp. Most food manufacturers will test their manufacturing environment for Salmonella and will halt production or recall product if Salmonella is even found on their equipment or contact surfaces or even down the hall in a drain. So this is why it's really important to monitor your workspace where your product is open to the environment, any contact surfaces, even areas that are further away. It's important to make sure that your sanitation program is handling those microbes by doing this environmental monitoring. Again, something that is really lacking in the cannabis space. Another point I want to draw attention to is that 1 spore does not equal 1 CFU. Aspergillus spores are also called propagules. They're not very viable. Most will not turn into a colony-forming unit when they are cultured, but that doesn't mean that they're not harmful. Many allergens are also associated with spores, so you can breed those in and have a bad health outcome, even if you don't get infected by those spores. So again, that's something that we have to keep in mind when we're talking about these inhalation risks. Both the fungal mycelium, so those are those thread-like hyphae that go through, the substrate that Aspergillus is growing on. That's where all the active metabolic processes are happening. That's where toxins are made and generated, and allergens are secreted, and spores are made on top of that mycelium. Both mycelium and spores produce DNA. So if you have DNA present for Aspergillus, it's going to ping on those PCR tests. So it's not just spores that are likely triggering these PCR tests. It's likely an active mycelium that was there growing on the product due to poor post-harvest practices. The California Department of Cannabis Control has done studies with cannabis, and they found in a publication back in 2022 that 100 Aspergillus fumigatus spores could ping a PCR test, but on a few occasions, those 100 spores didn't actually grow. So even though you had 100 spores, you saw no growth in your colony-forming units or your agar plates for viable microbes. So again, that illustrates that A spore isn't always going to be a viable CFU, but both can ping on your PCR tests. And that's really common. Spores, like seeds, since we work with plants and cannabis, they don't all germinate. So they don't all germinate into a living organism. There is no selective media that isolates only viable Aspergillus species. So unless you have a trained mycologist who can ID based on morphology, or you do a lot of comparative analysis and send out samples for sequencing, get their morphology lined up with the species that you're finding, and really train yourself, you really can't enumerate CFUs specifically for those 4 Aspergillus species tested for in cannabis. It is very challenging. And that's why total yeast and mold testing, or TYM testing, is so important, since these Aspergillus species will be part of those TYM counts if they are viable. Again, spores though, not many spores will grow. You're getting like a 1% viability out of those spores typically. But if you have high TYM counts and Aspergillus species on your product, the bottom line is you have a mold problem. I've only seen 1 or 2 cases where a grower failed Aspergillus but did not fail for total yeast and mold. The other, hundreds of C of A's that I've seen that show failures, usually when you fail for Aspergillus, you're also failing for TYM. So again, you have a mold problem. So in the end, my opinion, as a microbiologist studying Aspergillus in the lab, and not just studying viable Aspergillus, but also studying its bioactive components like allergens, is that we do need to test for pathogenic Aspergillus species, even if our current testing methods are not perfect. And there's also other species that we know infect people through the route of inhalation, like Cryptococcus neoformans, which is another fungal pathogen, and other Aspergillus species like Aspergillus versicolor, which are not tested for in cannabis, but also potentially are a health hazard through, again, that route of inhalation. So even though these methods are not perfect, and there could potentially be false positives, and there's this debate between viability and DNA presence, I still think that we should be testing for this. Now, should there be another layer that looks at more allergens, which we don't test for at all, or more toxins, which we only test for a handful of? I think that that's another potential avenue, or looking for viability through other types of agar media might be another way to kind of corroborate some of those PCR tests that we're getting back. Because the truth is that these microorganisms don't die when you combust them on your flower products or when you vaporize them. And that's a potential acute risk for living microorganisms and chronic risk to consumers with their toxins and their allergens. I also agree that we do have to understand better the inhalation risk to consumers with regards to opportunistic pathogens like Aspergillus. This means that scientists, medical professionals, consumer safety experts, and cultivators, we all need to come together on this and take it seriously. But the truth is, very little research is being done in this area. Until we better understand these risks, I'm an advocate for stringent PCR tests, especially since recent reports show that really only 4% of cannabis cultivators in Oregon are failing for Aspergillus. And even in other states, the failure rate for Aspergillus isn't incredibly high, indicating this isn't ubiquitous across all cannabis flower products, that it's likely linked, and especially in my experience, people are failing because of poor sanitation practices and improper dry cure, which allows these spoilage microorganisms, which Aspergillus is. It's a decomposer. It likes to chew up dead plant material, which is exactly what happens in post-harvest. It allows these conditions, poor sanitation and improper dry cure, allows Aspergillus to thrive. So, and then in addition, for folks who downplay the risks to recreational users, I just want to remind everyone that cannabis was first legalized as medicine for people who likely have compromised immune systems, and we need to figure them in as intended users and ensure product safety for them. For growers who are just throwing everything at their plants hoping that they don't fail for Aspergillus, it is your responsibility to ensure your indoor environments are free of pathogenic and toxigenic microbes in your air, water, substrates, surfaces, and other areas. Environmental monitoring and good sanitation practices will serve you much better in the long run than spraying with neem, PAA, H2O2, or remediating flour with ozone or X-ray remediation technologies. And I'll also add a cool video of an Aspergillus fumigatus biofilm that I grew in the lab. And I want to show you that visual, that Aspergillus is not just found as spores in the air, which I've been hearing that narrative a lot. It comes from a mycelium somewhere producing these spores. In your nutrient tanks, biofilms in your drip line, it colonizes your cocoa, and it lives in your dehumes. So it's not just one random spore floating around in your environment. It's likely that there's mycelium, that there's biofilms forming in other parts of your grow, and you really need to address those. I recommend that cannabis cultivators treat the big four Aspergillus species that we test for like the food industry treats Salmonella, Listeria, and pathogenic E. coli. If you put in those proper sanitation controls, you do environmental monitoring, and you're really making sure that you're verifying that you are reducing or eliminating these potentially harmful microbes out of your facility, that you won't fail for it on your final product. Good sanitation goes a long way.